Hi, and welcome to the Lab 10 Genomics Common Data as a part of EE542 Internet and Cloud Computing under Professor Young Cho. In this project, we will download the genomics, human genomic data, and try to learn from the biomarkers and predict the type of cancer that every patient has with an accuracy of 84% in this project, and the testing accuracy of 0.99%, almost 100% testing accuracy. The objective is to integrate pre-process, by filtering the data related to particular disease type and how we obtain the data from miRNA files. We extract the files based on the file ID and the case ID, and we get the metadata for each file and for each corresponding case. And we generate a matrix, the miRNA matrix, for all the files with labeled normal or with labeled tumor. Then we apply machine learning for the data to predict what kind of cancer that each case has. Before we start, before we dig into the project, let's see what a biomarker is. It's a measurable indicator which says the severity or the presence of a disease. There are three categories. 1, DNA methylation. 2, nucleotide variation. 3, transcriptome profile. The validation of data is performed on imported GDC and also NIC programs. To download the Genomics Data Commons, we need a data transfer tool. So the tool is provided by the NIC, which can download and also submit. So using this tool, we can download the genomics data and also submit in case we have some samples, which obviously goes through an approval process. It has a command line interface, and each time you download, you need to generate a UUID.
The step 2 of this experiment requires us to download the miRNA files. Let us go to this site, which is going to allow us to select some parameters, and we will be able to see the number of cases and the number of files. So these cases and files basically tells us the number of cancer, but sometimes it happens that some cases there won't be any cancer to the patient. So inside the cases, once we open the cases tab, we need to select inside the cases tab, we need to select the primary site. We selected all the options and the disease type. So in our experiment, we have selected all these options. Now inside the files tab, inside the data category, we need to select the transcriptome profiling. The data type is going to be miRNA expression quantification, and experimental strategy is miRNA sequence. After doing that, here I am generated with 11,486 files and 10,601 cases. So these are all the files which are generated after doing the necessary selections. Now, this next step tells us to click on the manifest download, and let me do that. After downloading this file, there are going to be 5 things inside this file, which is the file content ID, file name, the MD file, the size of the file, and also the state. The state basically tells you if the person is living or dead. Now, all these data that we see in here, we need it in our system. In order to get these, the GDC has invented a tool which simply helps us to retrieve all the data. So by running this command inside my terminal, I will be able to download, or I'll be able to get the GDC client tool. So this client tool, I've installed it or downloaded in or extracted into my system, and here it is. Now, moving forward, we need the data from the website. So I'm going to make a live miRNA directory. I will open it using the cd command. All right, so this is the directory which is created inside or opening this directory. I have all these files. How did I get these files? By running this command in here. If I run this command on my terminal, I should get a successfully downloaded. So this process has taken me about 2 hours. And after 2 hours, I was able to download all these files. So I've taken a screenshot to show you that it was successfully downloaded. So I have about 11,486 files. Now, we go inside the live, and these are the files which were downloaded, which is a good thing. Moving ahead, we will run this Python code, which is the check.py. This check.py, let me open it to show you what it does. It's a Python code. It's a very easy code. What it does is it is going to check the MD5 inside the files which are generated. So I have all the files inside the live miRNA folder. So inside that folder, it is going to match the MD5 with the manifest file. So remember I told you that the manifest file consists of an MD file column. So this MD file column, it's going to be matched with the files inside the live mRNA. Okay, so after running this command, which is the check.py inside the terminal, I am able to get a successful downloads, or the successful checking. Right. Now, moving forward, here, once we get the biomarker files, we need to get the case IDs related to their file IDs. And this is because we want to correlate the biomarker files with the corresponding biospecimen files. Okay, so in order to do that, I have a parse file case code, which is a Python code again. Before that, we would need a JSON file, which I have downloaded already. I'm going to show you inside this folder. Here you go. So this is the file which was downloaded. And let me show you what the code actually does. All right. So all it does is it matches the file ID with the case ID. And this is the input file which we downloaded. And this is the output file which should be generated if the code was perfect. So let me just run this code to show you really quick, because it did not take me much time. All right, so good to go. So it worked, and here is the case ID file which should be generated, and it is. It is a CSV file. So it shows that for 11,486 cases, the file ID was matched with its case ID. It's pretty straightforward. It will go till the end as I'm taking it. There you go. So this basically completes the step 9.
Get the metadata for all the file ID and case ID. In order to do that, we are going to run request meta.py. Here is the code for request meta.py. In the fields, I'm going to extract field ID, file name, submitter ID, category, tumor descriptor, tissue type, primary site, and disease type as it was mentioned in the email. I'm going to set the size in the parameters as 12,000 as there are 11,486 cases. This will take the file case ID dna.csv as the input and produce two output files, which will be files meta.tsv and cases meta.tsv, which is going to contain the metadata. The file case ID dna.csv is the output, which is created from parse code, parse file ID Python code. Now, this sometimes takes a while because it is getting the data for 11,486 cases. And as given the data directory, this is a path where we'll find the output file. After doing this, we are going to run gen mir in metrics.py, which is basically in order to generate the mir in a matrix with all the cases ID labeled as normal or with tumor. So if it is normal, it will have 0, and if it has tumor, it will be with the particular tumor type. Alright, so the code has ran and it is going to create two output file meta.tsv. On opening it, we get all the metadata and there will be 11,486. So the next step is to run gen mir in metrics.py. This is the code for it. So as shown over here, if it will be normal, the label will be 0, but if it will be with tumor, the label will be either from primary site or from the disease type, as I've loaded it in the data frame. And the input file would be file meta.tsv, which we got after running request meta.py, and the output file would be mirnmatrix.csv. This sometimes takes a while because it is getting the data for 11,486 files. And we will get the output of CSV file where there will be the file ID, the metadata, and the label. On finishing this, we will be done with part 1, and part 2 is applying the machine learning package to the above data. For that, there are the following steps which have to be followed, which would be: first, we'll standardize the data, after which we are going to split the data in training and testing set, and then feature selection, after which we are going to use the machine learning algorithm and evaluate precision, sensitivity, F1 score, accuracy, and specificity. Okay, so the code ran. We got 691 normal samples and 10,795 tumor samples. Okay, so if we go to label, we should be able to see the tumors which are extracted from either primary type or disease type. As we can see in the label column, it is different types of cancer tumors, and if the person does not have any tumor, it will be labeled as 0. The next step is to run predict.py, and here is my code. So the first step would be importing all the important libraries which we are using, which is Panda, sklearn, and then I'm using logistic regression as the machine learning algorithm. So from sklearn.linear model, I'm going to import that. We'll load the data in the iris dataset, which is mir in a matrix.csv. After it has been loaded, I'm going to factorize the label column. This is done because in the label column, it contains string, and we cannot train the data on string. So I'm going to convert all the 43 different types of tumors to corresponding numbers, and after that I'm going to train the data here. After that I'm going to divide into training and testing set, use logistic regression with solver as Newton CG. When I was using liblinear, the data did not converge, so I shifted to Newton CG and increased the number of iterations to 250. And after that we are going to plot ROC curve, and also to and use 1 versus rest classifier for that. And since it is a multi-class, I need to label the binarize. So on running this code, I'm getting a training accuracy of 1 and testing accuracy of 0.8392. And this is the receiver operating characteristic curve. And after plotting the evaluating matrix, we get precision, accuracy, FN score, recall as shown. And the scatter plots are is shown over here for the PCA. This usually takes about 30 to 35 minutes to run, which is why I have already executed the code on my system so that I can show the output. Here is the code for PCA implementation, and I've used Seaborn library as an pair plot to get the scatter plots showing variation per principal component.
We have uploaded the source code, presentation, the statement, and data on GitHub. The link is provided in the presentation and also the submission text box. It's also uploaded on Google Drives just in case. Let's look at the GitHub link right now. In this, we find the details with the README attached and the presentation, data, source code, and everything on GitHub. Once again, thank you for viewing the lab. We welcome all feedback. Please like, subscribe, comment, and share.